Temperate ecosystems experience dramatic seasonal changes in their environmental conditions. These seasonal changes result in well-known ecological processes such as leaf fall, bird migration, changes in fur color, and aquatic insect emergence. While some of these processes might take place over a short time, their effects on ecosystem can be measured over longer timeframes. Here, we focused on leaf fall in temperate ecosystems and the role of leaf litter across the seasons. Every year, temperate forests produce several billion metric tons of new leaf material. The vast majority of this production enters the detritus pool. This leaf litter detritus is a fundamental resource of carbon and nutrients for terrestrial food webs and for aquatic ones. Streams and rivers are in fact responsible for the transport and processing of a substantial amount of terrestrial leaf litter. And while leaf litter enters the detritus pool largely once a year, making it an emblematic symbol of the autumn season, this detrital material can move and be decomposed within and across terrestrial and aquatic ecosystems throughout the whole year. We can visualize how fluxes of leaf litter connect terrestrial and aquatic ecosystems as follows. Leaf litter can enter the terrestrial and aquatic detritus pool by direct fall from the trees, but it can also move from the forest floor to the stream via lateral transport. The leaf detritus can then be decomposed by either microbes like fungi and bacteria, or by higher consumers, here represented by invertebrate organisms. The research question we wanted to answer was, how do material flows and decomposition rates of leaf litter change across seasons in both terrestrial and aquatic ecosystems? To answer this question, we use leaf litter traps and decomposition assays. We measure material flows using vertical traps and lateral traps. We measure decomposition rates using successive litter bag assays. We measure both leaf litter flows and leaf litter decomposition across the whole year. As expected, we found that leaf litter entered the detritus pool in large quantities in autumn. In this season, however, decomposition rates were only average. In summer, on the contrary, flows of leaf material were low, but decomposition rates were the highest observed in the study. We therefore observed no synchrony between peak leaf litter availability and peak in leaf litter decomposition. Further, we observed that streams decomposed detritus at a much faster rate than forests did. In addition, microbes contributed more than invertebrates to the decomposition process. Lastly, we contrasted fluxes of recalcitrant and labile leaf litter. We observed inverse dynamics in the fluxes of these two litter types. Recalcitrant litter had high flows and low decomposition rates, whereas labile litter had low flows and high decomposition rates. If you would like to know more about our study, check out our report at Curr in Biology.